This coordinate line maps the mathematical arena for our analysis, the twisted Bird-map transfer operator, evaluated specifically on the interval from -3 to +3. Within this defined operator space, we test 3 distinct kernel families, the WBA, the Laurentian, and the alien kernels. The focus narrows further to a rigorous boundary subgrid, examined exhaustively in papers 37 through 44. This widened parameter window spans from 0.70 to 1.10. To study this continuous space, it must be discretized. Researchers project the operator onto a high-resolution Ulam grid, utilizing matrix densities of n equals 1024 and n equals 2048. Within this specific grid structure, the invariant density proxy collapses. It condenses from a distributed wave into a microscopic footprint. This collapsed state is classified as regime A. It is a mathematically certified and stable single-cell Dirac-like spike. The defining characteristic of regime A is its absolute isolation. Every adjacent discrete cell immediately surrounding this main spike registers a mass of exactly 0. This perfect spike creates a computational problem. Standard mathematical observation tools are engineered to measure smooth curves and gradients, not rigid, isolated pillars. Applying standard macroscopic diagnostic tools to this specific discrete system produces severely limited data. This dashboard illustrates the failure. Traditional global measurements for variance and oscillation flatline at 0 when applied to the spike, capturing no meaningful data regarding local behavior. The core observational tension lies in scale. Because these global metrics max out their utility at the n equals 2048 resolution, the massive primary spike obscures the horizontal grid base. The system becomes blind to subtle microscopic changes at the floor. Consequently, a pristine one-cell mathematical spike registers identically to a spike that is quietly bleeding mass into its immediate neighboring cells. To determine if the structure holds, we have to examine higher grid discretizations, pushing the resolution toward n equals 4096. The central question of paper 46 is whether we can prove the regime A spike structure breaks at higher resolutions without manually sifting through 1,000-dimensional density vectors. A newly engineered diagnostic toolset is required to magnify the invisible baseline data, bypassing the traditional global metrics. Finding a mechanism to read the local cellular bleed is the only way to verify if the mathematical spike structure survives the next resolution doubling. The primary diagnostic mechanism designed to solve this visibility problem is the spike complement map, denoted formally as Q of U. This chart demonstrates the first procedural step of the mapping operation. The protocol algorithmically isolates the absolute maximal cell in the vector and drops its value to 0. The second step takes the remaining microscopic data floor and applies an L1 renormalization to it. The resulting algebraic output is a high-sensitivity magnification of the proxy density's local structural anatomy. This magnified space is measured using 5 distinct scalar gauges, collectively known as the spike-quotient battery. The first key metric is entropy, or EAE. It calculates the mathematical spread and distribution of the remaining normalized density. The second key metric is boundary variation, or BV-COMP. This measurement isolates the structural edges and transitions within the remaining cluster. For a true unbroken regime A single-cell spike, the baseline readings for VOP, EAE, and BV-COMP all return exactly 0. However, if the input vector shifts from a single spike to a multi-cell uniform cluster, these specific numbers escalate. The spike complement map translates invisible microscopic structural changes into distinct numerical indicators. Paper 45 establishes the flagship lemma, detailing exactly how uniform K-cell clusters behave under this mapping protocol. The lemma identifies exact algebraic relationships for uniform clusters. Boundary variation equals 2 over k minus 1, and entropy equals the natural log of k minus 1. These finite-dimensional equations can be algebraically inverted to solve directly for k, the exact cellular width. This establishes a precise operational diagnostic shortcut. We can extract the exact cellular width of a cluster using only a single scalar measurement without ever accessing the underlying density vector. To stress test these theoretical formulas, paper 46 introduces the Path B experimental design. Researchers fed synthetic multi-cell clusters, adversarial probe kernels, into the system to verify if the algebraic inversions hold under scrutiny. This table captures the empirical validation data. It extracts the path B results for cluster widths of 2, 3, 5, 10, and 20. The experimental computer runs match the theoretical mathematical predictions to machine precision. Because the BV-COMP and EAE calculations follow separate algebraic routes, they act as built-in cross-validators. Agreement between the two confirms a uniform cluster diagnosis. This algebraic fingerprinting allows researchers to use these theoretical equations as a functional tool for decoding cluster width from a single scalar result. The ultimate operational purpose of this diagnostic toolset is establishing a strict falsifiability threshold for the series. The mathematical condition is binary. Any future ULIM run returning an EAE greater than 0 or a BV-COMP greater than 0 definitively breaks the regime A hypothesis. Across all 60 existing verified parameter rows evaluated at the n equals 2048 resolution, this threshold remains unbroken. But the Path A experiment executes a preliminary memory-intensive push to evaluate the operator at an n equals 4096 resolution. Researchers extracted specific preliminary stub data from the WBA kernel at parameter 0.78 at this high resolution. This run returned an entropy reading of roughly 5.31 and a boundary variation of roughly 0.823. The implication is immediate. The n-stable single-cell hypothesis is computationally falsified at that specific high-resolution frontier. Establishing this threshold provides a verified transition into regime B, proving the diagnostic battery works on live high-resolution data. To maintain rigor, we must define the strict academic boundaries of these claims. All structural findings and falsifications strictly apply only to finite-dimensional Ulam grids, not to the underlying continuum invariant density. Paper 46 introduces no new massive Ulam matrix calculations for the n equals 2048 baseline, relying on previously certified runs to test the probe kernels. Furthermore, the spike-quotient readings are finite-dimensional linear algebra measurements, independent of statistical conjectures or estimation. The comprehensive mapping of this newly discovered n equals 4096 resolution space is entirely reserved for the upcoming Paper 47. The validated spike-quotient battery now stands as a ready-to-deploy diagnostic mechanism at this new resolution frontier. Dynamical systems researchers now have an algorithmic mechanism to detect and decode the exact point at which the regime A spike structurally shatters.